Your exams don't do flap, neither do we. Straight talk without the sweet talk. All you have to do is listen, read, test, and destroy. Let's start this lecture now.
Let's walk through the bacterial cell envelope layer by layer, starting from the outside and moving inward. This approach mirrors how examiners test structure, function relationships in clinical stems. Every layer has a specific chemical makeup, a clear function, and predictable clinical consequences. When one structure is altered, it directly changes virulence, immune evasion, staining behavior, and antibiotic susceptibility. Bacterial structures function as an integrated system rather than isolated parts. Structure determines composition. Composition determines function, and function determines clinical behavior. This explains why changes in a single component, such as the cell wall or membrane, can alter immune recognition or drug response. Many questions test this indirectly by describing outcomes like resistance, sepsis, or failure of phagocytosis rather than naming the structure outright. Starting with appendages, flagella are long, whip-like projections composed of the protein flagellin. Their main role is motility, allowing bacteria to move through fluids, penetrate mucus, and spread along epithelial surfaces. Motility enhances colonization and dissemination, especially in gastrointestinal and urinary infections. Flagellin is recognized by innate immune receptors, triggering inflammation. Loss of flagella often reduces virulence. Examiners frequently link motility to spreading infections or ascending disease. Pili, also called fimbriae, are shorter hair-like projections made of protein subunits. Their primary role is adherence to host cells, which is the first step in infection. Without adherence, bacteria are removed by urine flow, ciliary motion, or mucus. Some pili are specialized as sex pili, which mediate conjugation. Conjugation allows plasmid transfer between bacteria. Plasmids commonly carry antibiotic resistance genes, virulence factors, or toxin genes, explaining rapid emergence of resistance without chromosomal mutation. Some bacteria form spores, which are dormant survival structures rather than reproductive forms. Spores contain a thick keratin-like coat, dipicolinic acid, calcium, peptidoglycan, and tightly packed DNA. They are metabolically inactive and resistant to heat, desiccation, radiation, and disinfectants. This explains persistence in soil, hospitals, and contaminated wounds. Spores revert to vegetative cells under favorable conditions, which is why exposure history and environmental clues are heavily emphasized in clinical stems. Moving to the cell envelope, the outermost optional layer is the capsule. Capsules are usually made of polysaccharides, except for one classic organism with a protein capsule. Capsules inhibit phagocytosis by blocking opsonization and preventing immune cell attachment. This increases virulence and allows bloodstream survival. Capsule presence explains why some organisms cause severe invasive disease despite a weak inflammatory response. Antibodies against capsule components are protective, which underlies vaccine design. Closely related but structurally different is the slime layer. This is a loose, unorganized polysaccharide coating that promotes adherence to surfaces and biofilm formation. Biofilms form on prosthetic devices, catheters, and heart valves. Within biofilms, bacteria are shielded from antibiotics and immune cells. This explains chronic, relapsing infections and poor antibiotic penetration. Examiners often describe indwelling devices and persistent infections rather than naming biofilms directly. In gram-negative bacteria, the outer membrane is a defining feature. It is absent in gram-positive organisms. The outer leaflet contains lipopolysaccharide or lipooligosaccharide. Lipid A is the toxic component responsible for cytokine release. This leads to fever, hypotension, shock, and disseminated intravascular coagulation. The O antigen is highly variable and antigenic, explaining strain differences and immune evasion. Porins are protein channels embedded in the outer membrane. They allow passive diffusion of small hydrophilic molecules. Changes in porin size, charge, or number directly affect antibiotic entry. Reduced porin expression is a common resistance mechanism. Many outer membrane proteins are antigenic and interact with host defenses. The outer membrane also functions as a permeability barrier, protecting the bacterium from bile salts, enzymes, and large antibiotics. Between the outer membrane and cytoplasmic membrane lies the periplasmic space. This compartment contains a thin peptidoglycan layer and numerous enzymes, including beta-lactamases. These enzymes can inactivate antibiotics before they reach their targets. The periplasm acts as a biochemical buffer zone, allowing rapid response to environmental threats while protecting the cytoplasm. Questions often test this by describing resistance despite drug exposure. The cell wall itself is made of peptidoglycan, which consists of repeating sugar units cross-linked by peptide chains. The cross-linking is performed by transpeptidase enzymes, also called penicillin-binding proteins. This lattice provides rigidity and prevents osmotic lysis. Differences in peptidoglycan thickness explain staining behavior. Disruption of cross-linking weakens the wall, leading to cell rupture. Altered penicillin-binding proteins are a classic mechanism of resistance and are frequently tested. Beneath the cell wall lies the cytoplasmic membrane. It is a phospholipid bilayer containing transport proteins, enzymes, and components of energy generation. Because bacteria lack mitochondria, oxidative phosphorylation occurs here. This membrane controls nutrient uptake, waste removal, and ion gradients. It also anchors proteins involved in cell division and shape maintenance. In gram-positive bacteria, lipoteichoic acids extend from the cytoplasmic membrane through the peptidoglycan to the surface. These molecules promote adherence and trigger inflammatory cytokine release. Their immune effects resemble endotoxin but are structurally different. Recognition of lipoteichoic acids explains inflammatory responses even in the absence of endotoxin. When comparing gram-positive and gram-negative organisms, the structural differences explain nearly all clinical behavior. Gram-positive bacteria have a thick peptidoglycan layer, teichoic acids, and no outer membrane. Their peptidoglycan is exposed, making them more accessible to many cell wall targeting antibiotics. Resistance often occurs through altered binding proteins or enzyme production, not reduced drug entry. Gram-negative bacteria have a thin peptidoglycan layer located within the periplasm, an outer membrane with lipidae and porins, and a more complex barrier system. The outer membrane limits antibiotic access, and periplasmic enzymes can degrade drugs. Lipidae contributes to systemic inflammation and septic shock. These features explain why gram-negative infections are often more severe and harder to treat. In summary, gram-positive bacteria resemble a thick exposed wall over a single membrane, while gram-negative bacteria resemble a layered sandwich with an outer defensive shield. These architectural differences explain staining patterns, immune activation, virulence, and predictable antibiotic responses. When examiners describe resistance, sepsis, device-associated infection, or immune evasion, the correct answer almost always traces back to one of these structural layers.